One of the most widely used experiments in student laboratories is on the heart function of Daphnia. In order to understand the way human cells, tissue, and organs work, scientists use model organisms in their laboratories. Often those organisms are not closely related to humans. Thus, they could be guinea pigs, rats, mice, hamsters, fish, and even worms and flies. We and those organisms all belong to the same kingdom, Animalia, and our bodies are made up of similar cells. Those organisms are used because it is easy to maintain and breed them in the laboratory settings. However, scientists have to run preliminary experiments before verifying which of those organisms will or will not suit the purpose of their investigation. Today you will run an experiment with Daphnia. Name comes from Greek Daphne, from the name of the nymph Daphne. The genus Daphnia includes more than 100 known species of freshwater plankton organisms found around the world. For example, there is such species as Daphnia cucullate, Daphnia magna, Daphnia pulex, Daphnia longispina, and many others. Frankly, we do not know what species of Daphnia we have in our experiment, so we will put the name as Daphnia sp. When the species within a given genus cannot be identified in a sentence, it can be replaced with sp. Sp should not be italicized. Today you will try to identify whether or not certain chemicals and conditions will affect Daphnia heart rate the same way they affect the human heart rate. Like humans, Daphnia belong to the same kingdom Animalia. But they belong to the phylum Arthropoda, a different phylum from us, humans. We belong to the phylum Chordata. Daphnia are often called water fleas because these planktonic animals resemble land fleas in their general appearance and jerky movements through the water. Real fleas are insects. Daphnia are crustaceans, sharing only an extremely distant ancestor with fleas. Its close relatives are shrimps, lobsters, crabs, and crayfish, not fleas. Both crustaceans and insects are arthropods, though. Daphnia are mainly freshwater animals with transparent exoskeleton. Many of them live as a plankton in lakes and ponds. They are too small and weak to survive in a strong current they are unable to swim against. Their food is life and death algae, bacteria, yeast, and decaying material. A Daphnia's heart is myogenic, meaning like in humans, its heart rhythm is generated by specialized muscles in the heart. Myo, muscle, genic, produce. So meaning it's originated in muscle tissue, we mean in contraction. To be more specific, contractile activity originates in the muscle tissue rather than from nerve impulses. As you perhaps know, in humans, adrenaline stimulates heart to beat faster, but acetylcholine and low temperatures slow it down. The Daphnia heart differs from human heart in many respects. In terms of the heart rate, the Daphnia's sinoatrial node is actually a collection of spontaneously active nerves in the body called cardiac ganglion. This means that it would be risky to extrapolate heart rate findings from Daphnia directly to humans without first validating the model. So the goal of our study will be to determine either or not the Daphnia's heart will react the same on the adrenaline, acetylcholine, and cold temperatures. What is your hypothesis will be? Will the Daphnia's hearts react to these chemicals and cold same as human heart or not? In our experiment, first we will measure Daphnia's heart rate in its natural environment, which is pond water. Then we will measure Daphnia's heart rate in acetylcholine to see if the heart rate will slow down like it does in humans when exposed to that chemical. Then we will measure Daphnia's heart rate in adrenaline to see if the heart rate will speed up like it does in humans when exposed to adrenaline. Then we will measure Daphnia's heart rate in cold to see if the heart rate will slow down like it does in humans when exposed to cold. And we will measure Daphnia's heart rate in room temperature pond water to see if the heart rate will restore to its original rate. The first and the last sets are our controls. When conducting an experiment, a control is an element that remains unchanged or unaffected by other variables. It is used as a benchmark or a point of comparison against which other test results are measured. Controls are typically used in scientific experiments. Without it, the experiment will not have any value. For this experiment, we will need to have one Daphnia. Yes, we will work with one Daphnia during the whole experiment, so you will have to handle it with care, making sure you are not killing it. We will transfer it from the beaker into the depression slide with a plastic pipette. Try to pick up the big Daphnia. It will be easier to work with. If the pipette opening is too small, cut it with the scissors. Just like this. Now you have a larger opening, you see? Right here. Make sure there is enough water in the depression so Daphnia can feel somewhat comfortable in there. Daphnia will constantly move in the depression slide. To restrain this animal, you have to use cotton fibers. Take a small piece of cotton wool and tease out cotton fibers. Then place them in the depression of your slide. Make sure that your Daphnia has been immobilized by creating a net or web of cotton fibers. Use prop to arrange them over or around the animal and let it rest for about 3 minutes. You will have to examine Daphnia under 4x objective, which will give you a total magnification of 40 times. The heat from the microscope lamp can have significant effect on the Daphnia's heartbeat. Therefore, close the microscope diaphragm by moving the iris diaphragm level located immediately under the microscope stage. It will also maximize the contrast, making it easier to see the Daphnia's internal organs. Keep the light on only while observing the Daphnia. Identify the heart of Daphnia, which is located on the backside of the animal, just above the gut and in front of the brood chamber. You will use stopwatch to time 15 seconds, and during these 50 seconds, you have to count the numbers of heartbeats. Since the heartbeat of the animal is very rapid, count the beats by making dots on a piece of scrap paper in the shape of S letter, or any other shape you prefer. Here's what I do. I think you got the idea. Count the dots and record the heartbeat rate as the number of beats per minute in the data table. First, you have to fill in only the first row, Daphnia A. Ignore the shaded part of the table for now. I will tell you later what you will have to do with it. By the way, how would you find the number of heartbeats per minute if you know how many heartbeats in 15 seconds? You are correct. All you have to do is to multiply the number of the dots you got on a scrap paper by 4. So if your number is 34, multiply it by 4 and you will have 136 heartbeats per minute. So you record the number 136 in your data table. Do not write the number 34. Don't have to. Not usually we have 3 trials in each experiment. This means you have to do the same things over 3 times. After you record the numbers that you got from the trials, you have to write the average number in the last column. What it will be? That is right, 136. You add the 3 numbers together and divide them by 3. The average will be used to build a graph. In pond water, Daphnia A has 136 beats per minute. Later, after you finish your counts, you will plot this number on your graph. The numbers you see now are random. I just used the first numbers that came to my mind as an example. Now you will have to get your real numbers. While you're looking in the microscope, your peer has to tell you when to start count and when to stop. For those who work at home, I will be such peer. Your microscope field is a computer screen. I will tell you when to start counting, putting the dots on the paper, and when to stop. Are you ready now? Get the paper and the pencil or pen. Ready? Start. Stop. Pause this video and record your first number. Those students who work in the lab, data for Daphnia B, C, D, and E will have to get from their classmates. Those who works at home will get this number from me on the end of this video. Let's get ready for our second trial. Ready? Start. Stop. Pause this video and record your second number. Let's get ready for our third trial. Ready for the third trial? Ready? Start. Stop. Pause this video and record your third number. Do not forget to calculate average of those three numbers you have. What we just recorded will be our control, the heart rate per minute in a pond water. Let's get ready for our second set. We are moving to our second table, acetylcholine table. Now we want to expose Daphnia to acetylcholine. In humans, this chemical slows the heartbeat, but forget this for now. When you record your data, you should not think about this. Do not make assumptions because it might bias your results. You will have to think about it after you finish collecting the data. For now, you simply behave as a recording device. Let's expose Daphnia to the solution of acetylcholine. For this, with the plastic pipette, you have to take pond water out. Be careful, do not hurt the Daphnia while doing so. Do not punch it with the pipette while taking out the water. Now quickly add the acetylcholine solution to the Daphnia. You have to work quickly. Here it is. Give the animal 1 minute to respond to the action of the chemical. Get ready to start our first trial with acetylcholine. Get the paper and pen or pencil. Ready? Start. Stop. Pause the video and record the number. Remember, you have to multiply it by 4 before recording it in the data table. Are you ready for our second set count? Ready? Start. Stop. Pause the video and record the number. Ready for the third trial? Ready? Start. Stop. Pause the video and record the number. Get the average out of your 3 recordings. Now we have to remove the acetylcholine and expose the Daphnia to the adrenaline. Take the plastic pipette and remove acetylcholine. Now quickly add pond water to the Daphnia to wash off acetylcholine. Do it fast so the animal will not get dry. Here it is. Give the animal 1 minute to rest in the pond water. Then remove pond water with a clean plastic pipette. And quickly add adrenaline. Let animal to react to the action of the adrenaline. Are you ready to record the bits by putting dots on the paper? Ready? Start. Stop. Pause the video and you know what to do now. Record what you have to record. And once again, are you ready? Ready? Start. Stop. Pause the video and record what you have to record. Are you ready for the third trial? Start. Stop. Pause the video and record your number multiplied by 4. Take the plastic pipette and remove adrenaline now. Now quickly add pond water to the Daphnia to wash off the adrenaline. Do it fast so animal will not dry out. Here it is. Give the animal 1 minute to rest in the pond water, then remove pond water with the plastic pipette. Add quickly cold pond water. Let Daphnia to sit in for 1 minute so it can respond to the cold pond water. And after 1 minute, are you ready to start? Ready? Start. Stop. Pause the video and you know what to do now. Record. Second trial now. Are you ready? Ready? Start. Stop. Record the number. Don't forget to multiply by 4. Ready for the third trial? Ready? Start. Stop. Make recordings in your data table. Take the plastic pipette and remove cold pond water. Quickly add regular room temperature pond water. Here it is. Let Daphnia rest for a minute or so. And are you ready for our final recordings? Ready? Start. Stop and record. Pause the video. Get ready now. Ready? Start. Stop and record. Pause the video. Get ready now. Ready? Start. Stop and record. Pause the video. And here all your 5 data tables where you record your data in the first row. Do not pay attention to other areas of the tables that are shaded. Those rows you will fill in by taking results from other students. Or if you do this not in the laboratory but at home, I will provide you with the data that I gathered myself from the students in the last semester. In the previous video on a metric system, you build a line of the best fit. In this exercise, you will have to build bar graph. Bar graph is a diagram in which the numerical values of variables are represented by the height of the lines of equal width. What you see on this side is just an example of the bar graph. Please do not ask me, do I have to record caffeine trial? By looking at this graph, you should get an idea how to proceed with your own data. Do not read into it. Your results may be very different from this graph. Here, the results from other students that carried out the experiment on the heart function of Daphnia. You will have to build the second graph using the averages of their data. But before you do this, I have to let you know that one of this group was cheating. Instead of doing the work, that group falsified the data. Unfortunately, sometimes dishonesty is found not only in the classroom, but even in the community of scientists. This is why some papers get retracted from scientific journals, including prestigious scientific publications. Not long ago, I, with help of my students, identified such falsification in a scientific journal. Here, looking at the student data, you have to identify which group cheated. Was it the group that worked with Daphnia B, group that worked with Daphnia C, group that worked with Daphnia D, or group that worked with Daphnia E? Identify the group of dishonest students by carefully examining the data. Disregard their data. Do not include it in your second graph. Finding falsified data will be 10% of your grade for this lab exercise. I give you a tip. To find the cheating group, you will have to do simple calculations. Think of how you found heartbeats per minute. After you have identified the group, you will have to explain on the paper how you found this falsification. Attach these findings and explanation to your lab report. To make a second bar graph, you have to finish fifth column of these data tables. Then average all the averages from each column. Build the second bar graph that will represent all the data collected by the students. Do not include the data from the dishonest students. Compare this graph with graphs that you built using your own data. Summarize the content of the data tables. Did you prove or disprove your initial hypothesis? Explain. Formulate your conclusion of this study.